otot kawan, tulang besi. Otot kawan, tulang besi.
Tak perlu menjadi hebat untuk memulai. Untuk berani memulai apa yang kamu impikan. Keluar dari zona nyaman dan jelajahi potensi yang kamu miliki. Saat kamu terhubung dengan orang lain, itu menjadi kekuatan untukmu. Saat kamu memilih menjadi UNISA, kamu menjadi lebih dari yang sebelumnya. Menjadi kekuatan baru bagimu. Di seluruh profesi, konsistensi adalah produk dari etos kerja kami. Kami membuka wawasan, pengetahuan, dan pengalaman yang mempersiapkan kamu untuk bertarung di dunia nyata. Di sini kami merubah harapan menjadi peluang untuk mencapai sebuah tujuan. Selama lebih dari 3 dekade, kami hadir mewujudkan cita-citamu sesuai dengan perkembangan zaman. Konsistensi kami di dunia pendidikan memberikan cara baru bagimu dalam memandang kehidupan. Mewujudkan cita-cita bersama kami adalah cara terbaik kami memprediksi masa depan dengan menciptakannya untukmu. Kami meyakini bahwa kunci dari kesuksesan adalah profesionalisme, standar kualitas yang tinggi, serta mengimplementasikan nilai-nilai yang terkandung dalam Quran sebagai pedoman yang dapat mewujudkan tujuan pendidikan yang bernilai duniawi dan ukhrawi. Di UNISA.
Selamat datang di Universitas Aisyah Yogyakarta. Universitas Aisyah Yogyakarta atau yang kita kenal dengan UNISA Yogyakarta merupakan kampus yang berwawasan kesehatan yang berbasis nilai Islam berkemajuan. UNISA Yogyakarta memiliki 2 kampus, kampus 1, dan saat ini saya berada di kampus terpadu UNISA Yogyakarta di Jalan Siliwangi atau Ring Road Barat nomor 63. Saat ini saya sedang berada di gedung garda depan UNISA Yogyakarta yang diberi nama Gedung Siti Walidah. Siti Walidah adalah pendiri Sopo Tresno pada tahun 1914 yang menjadi cikal bakal dari Aisyah. Di Gedung Siti Walidah menjadi pusat administratif. Selain itu, di halaman UNISA Yogyakarta juga memiliki halte bus loh. Halte ini memfasilitasi pegawai dan juga mahasiswa terkait transportasi dari kampus terpadu ke kampus 1 dan juga sebaliknya. Menjadi pintu masuk utama di Gedung Siti Walidah ini memiliki layanan terdepan seperti penerimaan mahasiswa baru, akademik, dan juga keuangan. Selain layanan akademik, Universitas Aisyah Yogyakarta juga memiliki layanan kesehatan. Seperti di belakang saya, terdapat klinik fisioterapi yang tentunya dapat diakses oleh masyarakat umum. Klinik ini juga menjadi penunjang pembelajaran bagi mahasiswa dengan fasilitas yang lengkap, sehingga diharapkan nantinya para lulusan dapat menangani dan juga memahami pasien dalam dunia kerja. UNISA Yogyakarta memberikan privilege kesehatan kepada seluruh pegawai dan juga seluruh mahasiswa dengan membuka layanan klinik dana sehat Muhammadiyah. Di sini, seluruh pegawai dan mahasiswa dapat berkonsultasi dan juga periksa kesehatan yang juga akan dilayani oleh dokter. Dan kalau kamu sakit, nanti akan diberi obat loh, gratis. Halo dok. Dan sekarang kita sedang berada di lantai 4 Gedung Siti Walidah. Di depan saya ini terdapat Hall Siti Barorah Barit yang berkapasitas 300 orang dengan setting tempat teater. Sehingga memberikan kesempatan untuk orang yang berada di belakang untuk bisa melihat ke depan dengan jelas. Di sini biasanya digunakan untuk acara himpunan mahasiswa atau kuliah umum. UNISA Yogyakarta mendukung penuh program Green Campus dan sebagai salah satu bentuk implementasinya adalah melakukan penanaman tanaman sayuran organik. Tempat ini terletak di samping Gedung Siti Walidah dengan memanfaatkan lahan tanah yang kosong. Sehat, sehat, sehat. Nah, menuju ke Gedung Siti Bariyah, kamu bisa berjalan kaki sambil menyapa teman-teman kamu yang ada di gazebo. Nah, di gazebo ini memiliki fasilitas seperti wifi dan juga stop kontak yang bikin kamu makin betah nugas atau nongkrong. Dan inilah Gedung Siti Bariyah. Siti Bariyah menjabat sebagai Ketua Aisyah pada tahun 1917 sampai 1920 dan terpilih kembali pada tahun 1927. Ini adalah auditorium Gedung Siti Bariyah. Biasa digunakan untuk acara besar seperti wisuda dengan kapasitas 1.000 orang. Di balik pintu berlapis timbal ini terdapat laboratorium radiologi.
Dilengkapi dengan alat X-ray sehingga mendukung mahasiswa untuk belajar dan memiliki pengalaman dengan alat yang sesungguhnya.
Karena menghasilkan radiasi sinar X, jadi pintunya jangan lupa ditutup ya. Ruang operasi? Bukan, ini skill lab operatif. Memiliki nuansa yang real seperti kamar operasi sehingga membantu mahasiswa untuk mendapatkan pengalaman yang senyatanya. Menjadi kampus berwawasan kesehatan dan inilah skill lab. Merupakan kompilasi dari berbagai ruangan praktek yang digunakan untuk mahasiswa berlokasi di lantai 3-6 dan bertujuan untuk menampung mahasiswa untuk melakukan praktikum. Nah, di sini adalah ruangan administrasi laboratorium yang terletak di lantai 3 Gedung Siti Bariyah Universitas Aisyah Yogyakarta. Sebelum mahasiswa menggunakan ruangan laboratorium, mahasiswa harus kesini dulu untuk konfirmasi jadwal dan juga peminjaman alat praktikum. Alat-alat yang dibutuhkan mahasiswa untuk praktikum sudah disediakan di sini dan juga sudah disiapkan per paket sesuai dengan mata kuliahnya. So, ini akan sangat membantu mahasiswa untuk mendapatkan peralatan pembelajaran media yang tepat. Kamu bisa mengakses balkon lantai 3 Gedung Siti Bariyah. Di sini merupakan salah satu spot foto yang menarik loh. Dan di belakang saya ini adalah Gedung Siti Munjiah. Siti Munjiah menjadi wakil ketua Kongres Perempuan pertama pada tahun 1928 di Yogyakarta. Seorang ulama perempuan yang sangat inklusif dan juga toleran. Nah, sekarang kita sedang berada di lobi Gedung Siti Munjiah. Memiliki 9 lantai yang mencakup ruang kelas, ruang meeting, ruang dosen, perpustakaan, dan juga perkantoran administratif. Ini bukan parkir basement mall loh. Tapi ini parkir basement gedung Siti Munjiah. Semua dosen dan tenaga kependidikan maupun mahasiswa bisa parkir di sini loh. Tapi jangan lupa parkir yang rapi ya. Berada di lantai 3 Gedung Siti Munjiah, terdapat perpustakaan Universitas Aisyah Yogyakarta yang telah terakreditasi A. Perpustakaan juga menyediakan fasilitas Aisyah Muhammadiyah Corner, digital data delivery, dan juga GoBook. Suasana yang santai dan juga nyaman dilengkapi dengan fun corner dan juga kursi beanbag sehingga menunjang mahasiswa untuk dapat berkunjung ke perpustakaan pada pukul 8 pagi sampai 3 sore. Dilengkapi dengan buku-buku yang membantu mahasiswa mulai dari bacaan ringan sampai yang membantu tugas, skripsi, ataupun tesis kamu. Dan seperti itulah kampus tour kita. Let's join us to be UNISA. Dah.
Be UNISA. Kami hadir sebagai solusi menciptakan generasi yang kreatif dan inovatif yang mampu berkontribusi pada pemberdayaan dan pencerahan, menuntun generasi menuju masa depan yang memiliki pengetahuan dan wawasan yang berbasis pada nilai-nilai Islam berkemajuan.
Health policies for a bright future. Health communication is us.
Professional Qurani for more productive, happier, and valuable life.
Health information technology, empowering healthier life.
Life engineering today, brighter Islam tomorrow.
Health architecture for more livable and hospitable building.
Wahai seluruh warga UNISA Yogyakarta, bulatkan semangat baktimu. Siapkan generasi unggul dalam prestasi, berjiwa profesional berani. Iman ilmu kuatkan, amal mulia kerjakan, akhlakul karimah wujudkan. Song-song fajar pencerahan tiba, sejahtera bumi bersama. Kembangkan sumber daya manusia, berkemajuan capai cita. Itulah misi kita bersama, sebagai sivitas utama. UNISA Yogyakarta, raih citra sempurna, sentosa abadi dan jaya. Ridho Tuhan selalu ku minta, anugerah limpahkan selamanya. Ridho Tuhan selalu ku minta, anugerah limpahkan selamanya.
Assalamualaikum warahmatullahi wabarakatuh. Izin masuk kawan-kawan semua. Memastikan bahwasanya untuk pembicara atau pematerinya sedang menuju via Zoom. Untuk itu dimohon kepada teman-teman semua agar bersabar dan tidak meninggalkan via Zoom. Terima kasih.
kuatkan, amal mulia kerjakan, apapun karimau wujudkan. Song-song fajar pencerahan tiba, sejahtera bumi bersama. Kembangkan sumber daya manusia, berkemajuan capai cita. Itulah misi kita bersama, sebagai civitas utama. UNISA Yogyakarta, raih citra sempurna, sentosa abadi dan jaya. Ridho Tuhan selalu ku minta, anugerah limpahkan selamanya. Ridho Tuhan selalu ku minta, anugerah limpahkan selamanya.
Bismillahirrahmanirrahim. Cek-cek apakah suara saya terdengar, teman-teman? Terdengar, Kak.
Terdengar.
Baik, terima kasih. Assalamualaikum warahmatullahi wabarakatuh. Selamat siang, salam sejahtera untuk kita semua. Shalom, Om Swastiastu, Namo Buddhaya, salam kebajikan. Yang terhormat, Bapak Wantonoro selaku Wakil Dekan 3, yang terhormat Dr. Azima Abdul Wahab selaku pemateri, yang terhormat Bapak Arif Yusuf Wijaksana selaku moderator, dan yang terhormat seluruh dosen Universitas Aisyah Yogyakarta yang sempat menghadiri pada siang hari ini. Dan yang saya cintai dan banggakan seluruh peserta yang sempat menghadiri juga. Alhamdulillahirabbil alamin. Puji syukur kita panjatkan kepada Allah Subhanahu wa Ta'ala, karena berkat dan izinnya lah kita dapat berkumpul di via Zoom kali ini. Salawat sampaikan salam, tak lupa kita hanturkan kepada junjungan kita, Nabi besar Nabi Muhammad Shallallahu Alaihi Wasallam, Nabi yang telah membawa kita dari alam kegelapan menuju alam yang penuh terang dengan artian kata minal zulumati ilan nur. Alhamdulillah teman-teman, pada sini kita masih bertemu lagi di Kuliah Pakar, yaitu Summertime 2022 Knowledge Reinforcement dengan tema Molecular Diagnostics in Virology One Day Collaboration with University Kuala Lumpur. Sebelum memulai acara, perkenankan saya, Kamilia Mamonto, selaku MC pada siang hari ini untuk membacakan susunan acara, yaitu pembukaan yang sudah kita lewati bersama, kemudian ada sambutan, selanjutnya acara inti, pemaparan materi, tanya jawab, dan terakhir yaitu penutup
Cek. Oh, maaf, apakah suaranya masuk? Masuk ya?
Oke, saya boleh dengar.
Oke, thank you. Thank you, Miss. Oke, Bismillahirrahmanirrahim. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, everyone. Welcome to Summertime Webinar Series 2022. I hope all of us always blessed by Allah Subhanahu wa Ta'ala and always healthy and always happy today. I think today this is a very valuable experience for us because it's first experience collaborative learning held by Medical Laboratory Technology Study Program Universitas Aisyah Yogyakarta with University Kuala Lumpur, Malaysia. Okay, before we begin, let me say hello to Indonesia student. Hello everyone. Bagaimana kabarnya mahasiswa dari Teknologi Laboratorium Medis Aisyah? Sehat ya? Banyak yang hadir ya ini. Oke. Ya. And then also greet your brother and sister, student from Malaysia. Hello everyone, how are you? Apakah ada yang dari Malaysia? Ada ya? Ya, oke. Before we begin, I want to introduce the speaker, Dr. Azimah Abdul Wahab, Head of Section Clinical and Biomedical Laboratory Science Sector, University Kuala Lumpur. The experience is a Doctorate of Philosophy in Virology, University of Surrey, UK, Bachelor of Science in Biotechnology, University Putra, Malaysia, and Matriculation College, Matriculation Malacca. Okay, hello dokter, assalamualaikum.
Waalaikumsalam warahmatullahi wabarakatuh.
Nice to meet you.
Nice to meet you too.
Oke, sehat ya dok?
Alhamdulillah sehat.
Ya.
Tuan pengacara sehat kah?
Ya, alhamdulillah.
Oke, alhamdulillah.
Ya, terima kasih. Kami amat gembira dapat bertemu dalam aktiviti kerjasama ini, dokter. Dan kami juga amat berharap pas majelis ini dapat diteruskan dengan kerjasama secara langsung antar institusi ya, insya Allah. Bisa bertemu secara langsung ya.
Ya, betul.
Oke. To shorten the time, I will read the topic of your webinar today. The topic is molecular diagnostic in the field of virology. We need to know that the pandemic has been running for more than 2 years. During the time, we have known the new science in virology and molecular, like as viral load, CT value, immune response detection technology, like as PCR, ELISA, molecular rapid test, and the other. To discuss it all and the detection technology in virology, I'd like to welcome your presenter to start the presentation. Please, Dr. Azimah Abdul Wahab, silakan untuk waktunya bisa mempresentasikan dan bisa berdiskusi dengan kita semua di sini.
Oke, terima kasih Tuan Pengerusi Majlis. Okay, Assalamualaikum warahmatullahi wabarakatuh and good afternoon. Okay, before we start with the lecture, this is an online lecture. First of all, I would like to thank for the invitation as the visiting lecturer through online class. So before we start, let me introduce myself. Okay, as mentioned by Chairperson just now, I'm Azimah Abdul Wahab, the head of sections for clinical and biomedical science section in University Kuala Lumpur. So here in UniKL, we have a few programs which under my sections, there are 2 programs that involve biomedical science and also medical laboratory technology. So let me give some briefing on myself. Okay, yes, as mentioned by Chairperson just now, my background is in the virology, and here in UNISA Tech, I teach both bachelor and also diploma level in biomedical and also medical laboratory science or medical laboratory technology. So I teach microbiology courses, basic microbiology, diagnostic microbiology, pathogenic, anything related to microbiology. But for today, as we know that you're going to see some sharing session from me that focus on the molecular diagnostic in virology. Okay, so hopefully this sharing session can enhance, can improve some knowledge about the basic understanding on virology as well as the molecular diagnostic that commonly used in a laboratory for identifications of viruses. Okay, let me share my screen. Okay, can you see my screen?
Yes. Okay. Okay. Okay.
Okay, so this is the thing that we are going to see later on during our sharing session. The first one is about viruses, and before we touch in detail about the technique, I think it's better for us to go briefly about virology, what the virus is all about their nucleic acid, their components, and also their replication process, because all this information can be applied later on in the indirect technique and also direct technique for identifications of viruses. So in general, for viruses is one of the microbiology study or field, which in the microbiology we have four major groups. We have bacteriology, which is study of bacteria, virology, study of viruses, mycology, study about fungus, and the last one, parasitology, which is study about helminths. So among these four groups, bacteria and viruses are the major groups that commonly associated with infection, either in humans or animals. So in general, I guess students in Indonesia and also in Malaysia also aware about the basic or understanding on the bacteriology, how these bacteriology, I mean how the bacteria replicates, how they reproduce. And then the identification technique for bacteria also is slightly different compared to identifications of viruses, because bacteria can be cultured in the on the what we call it artificial media compared to the viruses. So in general, for identifications of bacteria, you can culture them on certain types of culture media, either it's a selective or differential culture media, and also you can determine their morphology through the screening or staining technique. And then you also can see the sensitivity of the bacteria towards certain antibiotics and also their reaction against certain biochemical tests. So since bacteria can be grown outside of host, so it's easy for us to do the identifications of the microorganism. But for viruses, this microorganism, I would say can be categorized as non-living organism, because they don't have all the characteristics for a cell to properly function. If you still remember, I guess for a bacteria, we have a complex structure. They have a cell wall, they have the cytoplasma membrane, they have cytoplasms, functional organelles that play roles in the transcription and also translation process. But for viruses, they don't have all these components. That's the reason why they are not classified as living organism. It is called as particle, viral particle. So due to the simple structure or component of the virus, they are not easily identified, they cannot be easily observed using light microscope as what you can perform on the identifications of bacteria, because size of a virus particle is 1,000 times smaller compared to a bacteria size. So for virus, we need to use electron microscope to enhance or to zoom this particle of the virus to see the presence of virus in certain sample. And in terms of the replications of virus also, it's slightly different compared to bacteria. And for bacteria, reproduction, it commonly occurs through the binary fission, which one cell will reproduce, become two cells. Maybe it takes about 20 to 30 minutes for one cell become two, or maybe for certain types of microorganism or bacteria type, like a mycobacterium species, it takes a longer time, a few hours for one cell become two cells. So here we can see the different patterns of multiplications of this organism. Later on, I'll show you how viruses replicates in host cells. Okay, since this virus has no elements or components of a cell, so they need a host for replication. Because they are not cell type compared to bacterial cells, as long as they can obtain the optimum condition, like the presence of nutrients or maybe optimum pH, temperature, or maybe presence of oxygen, they can survive outside of host cells. But for bacteria, for virus, they need a host cells for replications of their nucleic acid as well as for translations of their RNA. Translations mean synthesis of their RNA. So we are going to see later on the process of the replications of a virus, depending on what types of nucleic acid carried by the microorganism. Okay, other than that, viruses also, even though they are simple structure or simple particle, not as a living organism, they still can cause infection in host. Different virus can infect a few types of hosts, but it depends on how they find the complementary proteins on the surface of the host itself, because the first step of viral replication is depends on the binding and attachment of the viral particle to the host. So similar like bacteria, viruses also can cause infection, maybe at your respiratory tract system, or maybe at your gastrointestinal system, on the skin, or maybe the other part of the what we call it membrane, mucous membrane area. And so here we can see that a few types of specimen or sample can be collected from patient for identifications of viruses. Okay, here is a structure. I guess all of students have seen this structure. Okay, basically a virus particle can have a capsid. Capsid is the outer part of the viral particle that play role in protecting the delicate inner nucleic acid in the viral particle. And so it protect the cells, so it protect the nucleic acids of the viral particle from any conditions like physical or maybe chemical reactions or any enzymatic reactions from the environment. Other than that, it also play role in the attachment of a viral particle to the host cells. I mentioned just now, every single type of virus, they have a specific binding protein on the host cells or host receptor. So they need to find the complementary proteins on the host system to allow their binding before they enter or absorb into the host cells. And here, this capsid has a spike protein. And this spike protein generated after the replications of virus in the host cells. So it play a role in many situations, including the attachment and binding of the virus into the host cells. Other than that is release of nucleic acid. This capsid also will decide which time is suitable for the nucleic acid to be released to the environment, whereas the locations, what time is supposed to be released to environment before the translations and also transcription process take place. Okay, then nucleic acid. Okay, in general, for a viral particle, for a species of virus, they only have either DNA or RNA. They cannot have both viral genome, either DNA or RNA. So that's the reason why some of viruses are classified as DNA virus and some of them as RNA virus, depending on what types of nucleic acid carried by the virus. So this nucleic acid and DNA or RNA can present in a single-stranded or double-stranded DNA or RNA. And also here, I give you an example of RNA genome, which is a calicivirus RNA. This calicivirus is a type of viral family that can cause different types of infection. Under this group or family of these calicivirus, there are a few species or strain like a Norwalk virus that can cause gastroenteritis problem. And here we commonly use what we call it, that Norwalk virus is a norovirus, norovirus type. It also one of the member for calicivirus. And another type of calicivirus is a feline calicivirus that can cause respiratory tract infection in cats and animal. So for this calicivirus group, any members under this calicivirus group, they have some similarity on their RNA genome or RNA nucleic acid. As you can see here, this RNA encodes, they have a sequence or gene sequence that encode for different types of proteins. Either that protein is non-structural protein, for example, polymerase or viral protease, and also structural protein, which is capsid. That proteins basically encoded at this open reading frame, ORF1, ORF2, or ORF3. For this ORF1, it encodes all non-structural proteins, including antipase, VPG, and also polymerase. Meanwhile, for the open reading frame 2 and 3, it encodes information for viral capsid. Okay, here, as you can see, this viral RNA has a protein that link at their 5' of nucleic acid. This is known as a VPG. And this is a viral protein that play a role in regulating host cells for their replications. And scientists have come up with the idea and also come up with their research work that this VPG basically can interfere the activity of host cells. And when the virus start to replicate the host cell, they try to hijack the machinery, the transcriptions or translation machinery in the system. So in the case of calicivirus, these VPG proteins, they take over the activity of translation machinery, which it has high affinity towards that system, the host system. So during the viral replications of this calicivirus, most of the machinery system proteins that involve in the translation process will be attracted to the VPG compared to the host system. So that is how they regulate, how they interfere the whole cells in order to allow synthesis of their proteins. Okay. And then some viruses have envelopes. Not all viruses have envelopes, only some of them. This envelope basically derived from host cells membranes. Once the virus leave or they complete their replication inside the host cells, and then they try to leave the host cell and pinch out together with the the host cells membrane. So that's the reason some of viruses have that envelope surround their capsid. So example of virus with envelope, or we call it as enveloped virus, is a coronavirus, the one that we are facing now, pandemic COVID. And then retrovirus, HIV virus, and also paramyxo viruses. Paramyxo is a group or family of virus that can cause respiratory tract infection. One of it is respiratory syncytial virus, and it can affect the functions of your lungs. And the next one is virus that presents without any envelope, and we call them as a naked virus. So example of naked virus is a calicivirus, picorna virus, and also adenovirus. So in Malaysia, I'm not sure either you're aware or not, from Indonesia, at the moment we are facing a problem with the increase of number on the HFMD, hand, foot, and mouth disease, which this kind of virus basically belong to the picorna virus group. So the number of cases seems increased, and inshallah everything will be okay in the first year, maybe after the next few weeks or months. Okay, so now we are going we already know the component of a viral particle. Now we would like to see how these viruses replicate, how they replicate in the host cells. Just now I've mentioned a little bit about the process, how calicivirus interpret the host system and how they replicate. Okay, calicivirus is RNA virus, but for the DNA virus, they have the other mechanism as well to hijack or to use the machinery in the host itself for the translations of their proteins. Okay, when we talk about replication, when we talk about the replication, basically viral replication can be divided into two categories. Either they are lytic cycle, and there are two types of cycle, lytic cycle or lysogenic cycle. So for that, lytic cycle is a general term that involves all stages of a viral replication, starting from binding of a viral particle to the host receptor. The interaction occurs between protein, spike protein on the viral particle that complementary to the protein of a host receptor. That's the first stage of viral infection, which is the binding process. And then the virus start to absorb, or maybe they enter into the host cells. And inside the host cells, they will replicate and to produce their proteins, their RNA, and then later on assembly and leave the host cells. However, another cycle of viral is a lysogenic cycle. That lysogenic cycle basically involve merge of viral genome with the host cells, with the host DNA, and allow the replications of the strand or genome together with the host cells until one time it become activated and will undergo the lytic process again. So here, as you can see from the slide, is a comparison between DNA and RNA virus. Okay, for DNA virus, any kinds of virus that carry DNA as their nucleic acid, once the virus bind to the host cells and then it enter into the host cells, they enter into the host cells. Inside the host cell, the capsid play a role in releasing all the nucleic acid. Okay, so here, as you can see, the viral DNA present in the nucleus of a cell. And inside the nucleus of a cell, DNA polymerase will play a role in replicating the viral DNA inside the nucleus. At the same time, the viral mRNA also will be transcribed, will be produced. So the importance of viral mRNA is for the synthesis of viral protein. So here the process starts with the binding of the virus, and then directly it goes to the nucleus for the transcription and also the translation process later on in the cytoplasm. But for this RNA virus, once the virus binds to the host cells, and then later on the viral nucleic acid will be released in the cytoplasm. Okay, at this cytoplasm, this is how the virus take over the machinery system for the translations. Okay, I've given to you just now an example, which is calicivirus, since they have the VPG at their 5' end of RNA. So what happened here, normally when any infections occur, your cells start to produce or synthesize proteins that are capable to control replications of viruses or control entry of viruses. So what happened during calicivirus infections, once they enter into the host cells, the presence of that VPG protein, and the protein that linked to the 5' end of the mRNA, they will have the high affinity to attract all the elements and all the machinery system like ribosomes, translation factors, and to their mRNA in order to translate their genes and to produce or to synthesize their own proteins. At the same time, they try to control synthesis of host proteins. This is how they regulate the infected cells. And then after the proteins is synthesized, and then one of the gene encoded by the viral protein is RNA polymerase. So this RNA polymerase synthesized from the translation process later on will start to replicate the viral RNA. Okay, and then the assembly take place at which all the what we call it, the nucleic acid will be carried into capsid to form a complete set of virus that we call it as a virion before they are released to the environment. Okay, sometimes mutation can occur during the replication process. The mutation commonly occur due to the activity of RNA polymerase protein during the transcription process and copying the nucleic acids of the RNA virus. That's the reason why most of cases involving mutation normally involve RNA viruses compared to DNA virus. Okay, next is identifications of viruses. So when we go to the identifications of viruses, I know you're also aware of what the common technique in the lab. Either it can be used as a technique for diagnosis of disease or it can be used for research purposes. So one of it is a microscopy techniques. Microscopic technique which we use electron microscope. A sample will be prepared on a slide. But this one need someone that has well-trained, someone that well-trained to handle the sample as well as to conduct or to observe the sample under electron microscope. Okay, another techniques for the identifications of virus is through the cell culture technique. Cell culture technique is a very common in the research area, which it provide or it facilitate the understanding for pathogenesis of a viral particle, because every type of virus, they have a different mechanism to cause infection in host. So by having the cell culture technique, actually it helps researcher to really understand how the virus itself can interfere the host cells and what they can do to control the replications of the replications of viruses in host cells. And then another technique for identifications of virus is through the serology, which is detections of antibody. Those who have been infected with any kinds of pathogens normally produce antibodies. So through the serology test, we can detect presence of the antibody in the sample or specimen. And the next one is the detections of microbial antigen. So these detections of microbial antigen will involve certain molecules or maybe certain substances, for example, antibody, to identify presence of the microorganism as well. And the last one is a molecular technology. Okay, so for today, I will cover this indirect detection and also direct detection for viral diagnostic. Okay, for the indirect detections. Okay, indirect detection involve the propagations of viral particle. This is known also as a viral isolation, meaning that we try to identify presence of the virus by looking at the maybe changes of the morphology of the host cells. And then we try to detect either the formations of CPE, a cytopathic effect, on the cells infected with viruses. So for indirect detection, there are a few techniques, which is the first one is conventional cell culture method. The next one is shell vial and also co-culture method, which this co-culture involve culturing two different cell lines that can support replication of viruses. And then for the indirect detections, it's also important for analysis and characterizations of emergent viruses, because by looking at the activity of virus in the cells, we know how to control the viral infection. And also it helps for the establishment of, if you want to design a new antiviral compounds, or maybe for the developments of vaccination as well. And the last one, in terms of the turnaround, the process takes times to produce a result. So it produces slow result turnaround. So it takes it depends on the species of virus itself. Sometimes for types of virus, it might take maybe 24 hours for you to see the CPE, cytophatic effect, the morphological changes of infected cells. But for certain viruses, it might take maybe a few days. For example, respiratory syncytial virus, you can start to see clearly the formations of the CPE after 3 days, after they reach the 72 hours of infection. Okay, the next one is a direct detection. So this direct detection means it detects virus directly from the sources. And the detection can be detections of nucleic acid or maybe detections of antigens through the immunological detection technique. So later on, I'll show you about the ELISA and blotting technique, which is one of the blotting technique is a Western blotting and also PCR. So for this one, PCR can produce a rapid result turnaround, but for the blotting technique, sometimes it takes maybe a few hours or maybe few days for you to get the final result. So normally for the direct detection, it's involve some cost, so it's quite expensive, and whenever it's involved use of antibody. Okay, now we go for the indirect detection. First is a traditional culture method, and this is a conventional method to understand the pathogenesis of a virus. Okay, as I mentioned just now, this culture method, cell culture method, commonly used in a research study. And when you would like to see what kind of mechanism used by viruses to interfere the host cells, so that you can come up with the idea on how to control the viral replication. So the cell culture technique involves seeding cells, seeding cell, or you seed the cells in culture plasmid. It's a cell culture plus, or maybe you seed cells in a well plate. We have a different types of well plate. We have 6 well plate, 12 well plate, 24 well plate, depending on what types of experiment you are conducting. And then the cells will be seeded in each of the well or flask, and then the cells also, the growth cells will be supported with the presence of culture media. As you can see from the picture, the red color will represent the culture media. We have different types of culture media, like a DMEM, we have RPMI. So in order to perform any cell culture technique, you have to know what is the best culture media that you are going to use to allow the propagations of virus in the cells. Okay, not only about the cell types, the cell line that you use for the propagation, you also need to know which of the cell line that can support or facilitate propagations of your virus. Not all virus can infect all types of cells. Right? It's quite difficult, basically, to find out which of the best hosts that can support the viral replication. For example, Kuliah virus, I would say norovirus. Just now I mentioned one of the example of the naked virus is a norovirus. Nowadays, study on the virus, norovirus, basically use other types of cells. Norovirus or feline Kuliah virus, they use other types of cell instead of the gastrointestinal, not gastrointestinal, the intestine cells, or maybe for the feline Kuliah virus, they do not use lung, even though we know that feline Kuliah virus can cause infection. So there's no lung cells or epithelial cells that can support for feline Kuliah virus. So what they do for the time being, to understand the pathogenesis of the virus, they use other types of cells. For example, that norovirus, researchers use murine, to study the norovirus, because since human norovirus cannot be cultured in the normal cells, so they use another type of norovirus known as murine norovirus. This murine norovirus can cause infection in murine, in rats. And then they use this murine norovirus as a model to study human norovirus, because this murine norovirus has about 90% similarity in their genome compared to human norovirus, because if scientists still looking for the best cell culture system to support the human norovirus. So they start with the murine norovirus first, and then culture them in the suitable host, and then allow the propagations of virus and see what mechanism used by the virus to allow the replications. And then once you know the right host for the replication, and then you start to inoculate your virus. So inoculations of your virus depending on your experiment. If you're conducting maybe molecular diagnosis, you already have the sample, then you can process your virus sample first before inoculate them in the cell culture. Or maybe if you're conducting research work, you already have your virus sample and virus stock, and you directly inoculate them in the well that been seeded with cells. And then you allow propagations of the virus due through the incubation period. Again, back to the types of the virus. Sometimes virus takes maybe around 24 hours for you to see the formations of CPE. Sometimes it takes a few days. Once you start to see the, meaning that you have to check day by day to identify formations of the CPE. And you can see the formations of CPE by using inverted microscope in the lab. Okay, here, as you can see, there's a result interpretation for the infected cells. The left-hand side, we have normal cells that been incubated for a few days. Meanwhile, for the infected cell, there's a morphological changes of the cells after a few days infected with viruses. So this is how we detect presence of virus in sample. Again, it takes time. So sometimes it's not suitable for the rapid diagnosis of the specimens from patients. Okay, and then scientists come up with another technique, which is enhanced virus isolations using conventional method as well. But this time around, it involved combinations, or they use another approach, which is by using a labeled antibody. The process is still similar. You still need to seed cells. Okay, for your information, the name is a shell vial technique. It's a shell vial technique, which is you use a vial, and then this vial also come together with a cover slip at the bottom of the tube, and then you will seed the cell, and then make sure it become a monolayer, cell monolayer. Later on, you will infect your virus into the cells. Okay, compared to the conventional cell culture method, the conventional cell culture methods allow the virus to naturally infect the cells, enter into the cells. But for this shell vial technique, we use centrifugation technique to enhance penetrations of the virus into the host cells. So we allow low centrifugation process, maybe around 1,800 RPM, or maybe up to 2,000 RPM for maybe 30 minutes. And by doing this kind of technique, the penetrations of virus or inoculations of virus into the host cell is much better and faster compared to the conventional method. And again, you need to incubate the sample. You need to incubate the sample, and then later on you will check the formations of CPE. It depends. Either it takes days or weeks for the formations of CPE. But to enhance the process, to shorten the process for the detections of virus, scientists has come up with the pre-CPE detections. For this pre-CPE detection, it detects virus post-cell entry before CPE present. And compared to CPE, and it's just based on your observation, morphological changes of the cells. And then if that CPE appear maybe after 3 days, then you can start, you know that, okay, this is the sample basically contain viruses. But for the pre-CPE, you can detect the virus post-cell entry, meaning that the CPE will not present at that time, but you already can detect the presence of the virus in the sample. So in this case, we use a labeled antibody. We use a labeled antibody to detect presence of the viral particle. That antibody normally specific to the viral antigen. So in certain cases, we use like antibody that's specific to the capsid of the viral. It's an example of viral antigens. Okay, and then for result interpretation, that shell vial technique, you can see from the results, the positive shows fluorescence color, which appear as like a yellow to greenish, green color that indicate presence of virus. Meanwhile, for the negative result, there's no production of fluorescence colors there. So we can identify presence of the virus even though there's no CPE present at that time. Maybe it just take for 1 or 2 days for you to detect the CPE. Okay? So it shortened the process of the observations of the CPE. Okay, then what is the limitations of culture technique for identifications of virus? Again, it depends on the detection system. And sometimes there's an absence of detection system for the agent, and also inappropriate cell culture system. As I explained just now, not all viruses can infect all types of virus. We need to identify the best one that can allow propagations of virus into the host cells to produce a new variant. And then some viruses also are categorized as fastidious virus. They cannot be cultured in the cell culture system. And then in order to understand their pathogenesis, then you have to use maybe animal study, or maybe you can use a tissue culture system to study more about their mechanism. And then a negative viral culture also from the cell culture technique does not mean that the agent is absent. Sometimes number of virus particle or maybe the titer of the virus is very low that can cause you get the false negative result. And the last one, not suitable for rapid diagnosis of pathogen in cases of extremely damaging or virulent viruses. For example, in the COVID-19, we need something that can produce result as soon as possible so that we can come up with treatments, best treatments for the patient to prevent the infection become severe. Okay, now we go to the direct detection. Okay, most of the technique for the direct detection basically use antibody. So here we'll start with the immunoassay based viral diagnosis. Diagnostic, which we use antibody for detection. It can be monoclonal antibody or polyclonal antibody. So there's a difference between polyclonal antibody and also monoclonal antibody. So in terms of the price, polyclonal is more cheaper compared to monoclonal. Because that monoclonal antibody is more specific. And when you use that monoclonal antibody, it can directly bind to the specific antigens or targeted area. Compared to polyclonal antibody, it has wide range of the binding site. And then detections of distinct regions on the target node pool, and then the applications of immunoassay based viral diagnostic can be applied for the serology study, which we can identify patients that have been exposed to a virus, and also it can be used to identify virus associated antigens from sample. In some cases, they still use a cell culture method. For that cell culture method, they can detect the virus associated protein within a cell line. But again, that technique will take it take longer time for the productions of result. One of it is the shell vial technique. Even though that shell vial technique is a modification of conventional cell culture technique, but again, due to the durations of the process, is still considered as something that will take longer time for you to produce a result. So scientists have come up with a few approach to enhance the process so that you can detect the viral particle as soon as possible. So what is the other approach? Is development of antibody-based assay to detect the target virus directly from sources. Okay, here is the ELISA, which is enzyme-linked immunosorbent assay. So in general, ELISA has a different types of format. It can be direct, indirect, competitive, and also sandwich format. Okay, for the direct and indirect, it has, for initial part, it has the same practice or the same mechanism, which we have to put the antigens on the surface of the ELISA plate. The coating process can be conducted in a few techniques, either through the passive absorptions or maybe through the chemical link to the surface. So you coat the viral, you can coat the antigen first on the surface of the ELISA plate. It can be applied for both direct and indirect ELISA. However, for the direct ELISA, you can directly add the primary antibody that been labeled with a certain component. So that component later on can give you a signal, and when you apply substrate, for example, for example, if you use chemiluminescent substrates, that chemiluminescent substrate will produce signals. It will react to the tag antibody and primary antibody to produce signal, and that signal can be detected by using machine. In this case, it can be detected by ELISA machine. Meanwhile, for the indirect ELISA, it applies two types of antibody. The first antibody is unlabeled antibody. They don't have any label on it, but that primary antibody can directly bind to the antigens on the surface of ELISA plate, and then followed by applications of the secondary antibody. So this secondary antibody labeled with a certain tag, and then later on it will produce a signal when you apply chemiluminescent substrate. Okay, that is about a direct and indirect ELISA. So the purpose for this direct and indirect ELISA is measuring antibody that responds against a given antigen. So in a virology, we can confirm if someone is having antibody against virus, targeted virus that we expect from the sample. Okay, next we go for this is a drawbacks. Before we go to the sandwich ELISA and also the competitive ELISA, we can see the drawbacks of the direct and indirect ELISA technique. Okay, normally for the detections of sample, detections of pathogens from sample, we will use real sample. That real sample might be contain different types of protein, numerous type of proteins. So that protein can cause a competition. They will have a competition to bind to the antigens on the surface of the, either to the antigens on the surface of the ELISA plate or to the surface of the ELISA plate during coating process. So it may cause the binding of non-specific proteins on the surface of the ELISA, which later on when you try to detect the signals, it can lead to the quantitation challenging. And we have no idea either that signal come from the antibody that directly bind to the antigens, or maybe it comes from the non-specific binding of proteins, other types of proteins. Okay, so how to overcome this issue about the non-specific binding of non-specific protein? Then scientists come up with this sandwich and competitive ELISA. Okay, so for this competitive ELISA, it's a detection of a viral protein directly from the sources. If you have maybe a blood sample or maybe you have a stool sample, then you can apply this competitive ELISA. So for this competitive ELISA, we will immobilize the targeted protein to the ELISA well, and target protein to the ELISA well, or as an antigen, ELISA well, and then we apply our sample. That sample will be mixed together with the antibody. So here we can see there is a competition between antigen in your sample and also target proteins on the surface of the ELISA, that they compete together for the binding of the antibody. So later on you will wash the systems to remove all the bindings of antigens and also the antigens and also the antibody, the level antibody. You remove all of them, and then we will detect later on the bounded proteins, and also we will detect the antibody, the tag antibody that link to the antigens on the surface of the ELISA. So normally for this one, it will produce signals that inversely proportional to the amount of analyte in the system. Because if your sample that you added previously contain antigens, and then it's already bind to the antibody that you have added together previously, and it will cause the concentrations of the antibody reduce later on. And then you can check the numbers and the inversely the reduce than the one that you have added at the initial part. And then we have sandwich ELISA. This sandwich ELISA is the best one because it's involved two specific antibody. So here we start with the coating of the capture antibody. And this is a specific antibody. That antibody can capture specific antigen from your sample. And then later on, we will add our specimen. So if our specimen for our sample contain the antigen that specific to the antibody that being coated to the ELISA plate, it can have a direct binding to the antibody, or it will be captured by the antibody itself. And then you will apply the primary antibody. And the rest of it is similar to the indirect. You will apply in primary antibody and then followed by secondary antibody. That secondary antibody normally labeled with a tag that can be identified by using chemiluminescent substrates, for example. So to detect the signal, then you can use ELISA machines. So for this one, sandwich ELISA, it more specific, is higher specificity and the more sensitive ELISA format compared to the rest of them. Okay. So for this ELISA, we have seen the different format, the different step or mechanism that we can perform for identifications of virus. So, however, there's some also drawbacks of the ELISA technique. It may increase the risk of cross-reactivity of antibody to other co-infecting virus, which can lead to the false positive result. And also, if you have low concentrations of virus in a sample, most probably due to the conditions that the sample was collected from patient that at early stage of infection. Maybe you have a very low virus titer in the sample, and then you couldn't detect presence of the viral particle from the sample. It may lead to the false negative result. However, the advantage of performing this ELISA, you can see that this ELISA comes in a multi-well layout, meaning that you can perform numbers of samples at the same time, so that you can have numbers of results once you have conducted or performed that experiment. And it can be conducted simultaneously. So this is very important during epidemic and also pandemic scenario to fasten the process of identification of virus. So here is a ELISA technique. Do we still have other options for detections of virus? Yes, and we still have a few more. But for today, I'm not going to cover all of them, just something that common that we can practice in the lab or maybe for the research purpose. And the next one is through the blotting technique. The blotting technique, the mechanism, the fundamental idea is kind of similar, like direct and also indirect ELISA, which you will bind or you will coat your specific proteins on the surface. Like an ELISA, you do the coating process for the direct indirect on the ELISA plate AC. But for the Western blotting, for example, in this case, I give you the example, we will attach sample on the membrane, a specific membrane. Either you can use cellulose membrane or maybe you can use PPDF. For Western blotting, the most common membrane that we use to allow binding of the proteins to the surface of membrane is a PPDF membrane. And then here, just imagine this is the membrane, and then you have your targeted protein from your sample, and then you can add a primary antibody, and then later on enzyme conjugated antibody, and then you will detect the signal by applying calorimetry or chemiluminescent substances. Okay, so in terms of the process of Western blotting. Okay, this Western blotting, in general, for research purpose, it takes a longer time, and longer time, it takes maybe a few days for you to get the final result. So in general, for students, what we commonly use in the lab, this is one of the things that I commonly use in my lab for my research work, because me and my team, for the time being, we are looking for the activations. We are looking for the activity or activations of certain protein or kinases in the cells infected with viruses. So we would like to see what kind of protein activated or maybe suppressed during the viral infection. So everything is performed using this Western blotting technique. It takes a few time. It takes a few days sometimes, because we are doing research work. So before we have the virus sample, we need to do the infection first in the cell culture system, and then you do the isolations of a virus, or maybe the isolations of protein from the infected cells. And then once you have isolations of the viral protein, just in case in here, if you want to detect the viral capsid, for example, you have to do the virus isolations from infected cells. There are a few techniques you can apply. That's the reason why I said for today it's more to the theory. So it's quite difficult for me to explain in detail the process. But I try my best to give you the idea on how the Western blotting technique is performed. You will do the virus isolation first in order to get the specific protein that you would like to see. Okay, there's a few techniques that you can conduct for the isolations, and then once you have that protein, we suspect that protein maybe come from your infected cells. Okay, then you have to do the lysis process of your sample. So this process involve need to be handled at a very low temperature because we are dealing with proteins. So normally any sample that involve protein sample, protein content must be conducted on ice, and to ensure that the ice will not be denatured. So everything must be in the low temperature to maintain the protein conditions. Okay, we conduct the lysis of cells, and then we will collect protein. Once you have that protein later on will be loaded in the SDS page. SDS page is a system that allow movement of protein throughout the gel. Okay, either you can purchase is a precast gel, and you can purchase directly from supplier, but quite expensive, and because it's a more clear items. But if you have the knowledge on how to prepare the gel manually, basically it's a simple test. No, it's not a simple test, it's a simple technique. You have to prepare gel based on based on the weight of your protein. If your protein is a kind of large protein, maybe you need to prepare gel with concentrations of SDS around 4 to 5%. And large protein can be more than 100 kilodalton protein. And because when you run the SDS page, normally small molecule of protein or maybe a low kilodalton of protein will move faster compared to the large kilodalton protein. So there's some you need to identify first, do the optimizations, which is the best SDS page concentration that you are going to prepare. For detections of large protein, maybe above than 100 kilodalton, normally here we use around 4 to 5% of SDS page. But sometimes we need to identify small protein maybe the size of protein around 15 to 25 kilodalton. And if you use 4% of SDS page, within a few minutes, you will lose the proteins from your system. So we need to increase the concentrations of SDS page for low molecule of proteins, with low kilodaltons. So for 20 to 20 plus kilodalton of protein, normally we use around 17% of SDS page, and then it allows the movement of protein slowly through the system. So since it's a small protein, it will slowly move downward. Maybe it takes about 1 hour for the cells to, for the protein to reach maybe the middle part of the cells, or maybe 3/4 from the bottom of the gel. So we allow the separations of protein. So that's the importance of the functions of SDS. We try to separate the protein based on their weight. And then when you do the separations of the protein, that separation is conducted using the electrotransfer, Western blotting machine. The one that you can see from the system, this is a Western blotting technique. You will run the machine, and then it will do the separations of protein based on their weight, and then later on, that protein from the gel will be transferred onto PBDF membrane or maybe cellulose membrane. It depends on what types of membrane you have. So since we have separations of protein, separated protein, you transfer them onto PBDF membrane, and then your protein later on will be coated ataupun present on the surface of the membrane. This is the first part. And you can see it takes a longer time for you to coat the sample on the surface of PBDF. And then later on, you will start to apply your specific antibody, which this normally specific antibody is an unlabeled antibody. It can, let's say, if you want to detect viral capsid protein, you must use antibody that specifically bind to the capsid protein. And then later on, use the secondary antibody and label with the enzyme conjugated and apply chemiluminescence. And that chemiluminescence will cause reactions with the enzyme and produce signals. So how can we detect the signal? Detections of the signal can be done using imager. They will detect productions of the signal and then come up with the result, as you can see here. This is how the Western blotting result is interpreted. So in terms of the cost, it's a cost effective, and you can apply small sample volume. You can detect multiple sample at one time, because at the SDS page, when you prepare the gel, basically you will have a few well for sample loading. So here we have a different wells that can allow movement of different sample in each well. You can see the different well. However, it only can provide you with qualitative result. You can see either the level, there's a presence of band or not on the sample, which indicate presence of the antigens, or maybe if you want to see the trend, either it decrease or increase during the infections, also represented by the qualitative result. But if you want to do the quantification technique, then you have to perform another technique as well. So it's a slow turnaround, takes a few days, and not the best technique for the rapid diagnosis, I would say. Okay, next one is lateral flow immunoassay. So this lateral flow immunoassay involves detections of virus associated protein directly from source. So the application is almost similar with the sandwich or competitive immunoassay, and you can read the results by the color changes at the test line. So I think each of us, I guess each of us have experience to handle this kind of kit, especially with the COVID pandemic. This is kind of self-test kit that you can apply. You can collect your own sample Either you can use a nasal swab sample or maybe a saliva sample. That sample will be loaded in this sample well, and it allow movement of the sample in the strip, inside the kits, and you can check either there's a presence of antigens or not by looking at the formations of the band. Okay, this is how it works. Okay, let's say if you load your sample at this part, and that sample contain analytes and also your specific antigens. And in this part, there's a presence of labeled antibody. This labeled antibody is specifically designed for your specific target proteins. And then if there is a binding interaction between the analyte, the specific target, and also the antibody, it will move through the capillary reactions, and there's a binding between these two. Later on, when it comes to the end of part, you can see the formations of band. If it's positive, you can see the line, because at this stage, you can see productions of a color on the test kit. This color basically can be produced by using a gold nanoparticle, or maybe you can use carbon nanoparticle or a colored latex bit, something like that. So the presence of this molecule can be used for detections of results using your naked eyes. You don't have to use something sophisticated machine for identification of results. So this is how it looks like. For positive result, it appears as this one. This is a very clear band, and this one is kind of blur, but it's still there. Maybe the titer, I would say the titer is quite low, but still can see the double band here. But for the negative result, you only can see a band at the control part compared to the test area. So for this lateral flow immunoassay kits that commonly available in the market, it has a fast turnaround. You can get result within minutes. And in terms of volume of sample or specimen, it only require very small volume. It's a cost effective. Even for the COVID-19 here, we can purchase the self-test kit maybe around 5 ringgit to 10 ringgit. It's quite cheap nowadays. And then it allows simple observation. You can observe the result directly using naked eyes. Easy to use. Anyone can handle the kits. We don't have someone with the proper training to conduct the study or analysis. However, this one just provide you with a qualitative result. It provide you qualitative result, not quantitative result. That's the reason. If positively confirm using this self-test kit that you are having positive COVID-19, to further confirm either you're positive or not, then you can go for the RT-PCR if you're infected with the COVID-19, for example, for the confirmations of the pathogens. Okay, now we go to the last one for the nucleic acid based detection method. Okay, this is a technique to detect viral nucleic acid. To detect viral nucleic acid in the specimen, and it utilizes multiple stepwise temperature cycle and also polymerase enzyme to amplify the DNA strength. Because during the process, sometimes the temperature will be increased and sometimes it will be lowered. It depends on what the stage of the cycle. And a standard polymerase used PCR can only synthesize from a DNA template. That's for the standard PCR. If you are infected with the DNA virus, then you can use the normal or standard PCR. But if you are infected with RNA virus, we need to there's an additional step here, which is involve transcriptive enzyme. This transcriptase enzyme is used for the amplifications of RNA virus. So here we have two types, PCR and also RT-PCR, which is reverse transcriptase PCR. And for the real-time qPCR, it can measure the productions of the target throughout the reactions. It can quantify the number of products. So here we have a viral RNA, and also let's say here if we have cellular DNA, or you can take it as a DNA, viral DNA. So for the RT-PCR, you have to what we call it, we have to amplify. And before we amplify, we have to prepare the template for the system first by using the reverse transcriptase enzyme, and then it's followed by the actions of the polymerase, and then followed by the rest of the steps. So this is the process before you proceed with the PCR. Okay, the first one is isolations of viral RNA. Okay, basically, if we refer to the COVID-19, COVID-19 is a RNA viruses. So once you collect sample from patient, or for suspected person, then you have to do the isolations of the viral RNA. So the isolations of this viral RNA, for example, involve the denaturing process. It involves the lysis buffer, and also inside the lysis buffer also contain the RNase inhibitor. And this RNase inhibitor is important to protect your RNA and also to ensure that you can isolate intact viral RNA. So once you do the lysis process, you will start to do, maksudnya once you do the lysis process, you will get separated RNA and also other content from the sample. And then you start to do the separation, separations of purification process. So in this case, for the at this stage, the nucleic acid, the sample will be loaded into the spin column. So it will adopt the centrifugation process. So in that column basically has the solid phase, like what we call it, the solid phase. I can't remember the name. Sorry. It has a solid phase to trap the RNA, presence of the nucleic acid, the RNA. And then you allow the centrifugation process. So at this stage, all the molecules other than RNA will not be retained, will not be retained in the system, and then will now be passing through the system. And it just let the RNA bind to the solid phase. And then later on, you will perform the purification by washing the spin column. And the washing step involves washing technique. And again, at this stage, we will wash your sample in the column and remove any excessive impurities from the sample. And it just leave your RNA. And then later on, you will add the elution buffer. So the elution buffer is important to remove your RNA from the solid phase in the system, and then it will be collected in the new sample. Meaning that once you have performed the washing step, you centrifuge your sample, remove any impurities from the sample, and you have that pure RNA, then you can transfer that column, spin column, into new centrifuge tube. At this stage, you can apply the elution buffer so that the elution buffer can remove your viral RNA from the solid column, immobilization column, and then you will collect your sample or RNA sample that will be amplified later on using RT-PCR. So for the RT-PCR, you need to prepare the reaction mixture. Reaction mixture. That reaction mixture contains transcriptase enzyme. It has nucleotides, primers, and also Tegman probe. You have to follow the reaction. You have to follow the instructions on how to prepare the reaction mixture, and then mix together with your RNA sample, and then you do the amplifications of your RNA using the RT-PCR. Okay, here the versus between RT-PCR and also PCR. For PCR, it can directly do the denaturation process, which is separate to the double strands of DNA, and then the annealing process, which is binding of the primer to the prime end, will initiate the extensions of the strand by adding in the nucleotides, and then the process repeat and repeat again. For the RT-PCR, you have to do the reverse transcription first by using the reverse transcriptase enzyme, and then the process will follow as what a standard PCR process. So the detections of your product can be performed by using gel electrophoresis, which once you have your sample or your product from the PCR, then you can load your sample in the gel electrophoresis in the gel and conduct the gel electrophoresis process to detect presence of the antigens, because we can detect the fluorescence color from the product as well. Okay, that's the general idea about the technique, common technique or conventional technique, as well as a molecular technique that we can perform in the lab for detections of viruses. So basically, for the molecular study, for detections of viruses, especially when you work with the molecular study, it cannot depends on the theory parts only. We need to do the practical part to improve ataupun to enhance our knowledge, our understanding in the technique as well. I know by having these sessions, lecture sessions, we can only understand the basic understanding, the basic idea about the molecular study. But when we perform it in the lab, then you know better on how to perform the technique. So that's the end of the topic. But before we go to the next step, which is a Q&A session, I just want to share to you about general information about UniKL. Again, UniKL Mestek and Medical Science Technology is one of the branches of UniKL campuses. And in Malaysia, we have around 12 campuses, including one more institute of postgraduate study. So these campuses has their own niche. They have their own specialization. For example, UniKL, we met at the top one, it more focus on the marine engineering. And then we have BMI that focus on electrical, electronic, aviation. One of the campus focus on the business, entrepreneurship, on the chemical and bioengineering technologies, and a few more. So our campus more focus on the medical science technology. We have 2 programs here related to the medical science technology. But as a whole, in the UniKL Mestek, we have 3 bachelor programs, which are Bachelor of Biomedical Science, Bachelor of Environmental Health, and also Bachelor of Occupational Safety and Health. For diploma level, we have a Diploma of Medical Laboratory Technology. So today we have our students with us, DMLT student, they are final year student. And also we have Diploma in Environmental Health, and the last one, Diploma in Medical Assistant. If you want, let's say, I'm not sure, I guess you are in the bachelor program. Let's say if you have intention to further study to the postgraduate level, here we offer Master of Applied Bioscience. So any programs that related to the science, or maybe biological science or medical science, then you can apply the postgraduate programs if you are interested with. So UniKL Mestek also organize public seminars, trainings, and also conferences. And sometimes some of our members or staff in the UniKL also provide consultancy services to the public. So this is latest information that I can share with you. At the end of this year, on the 23rd to 24th November, our campus will be organizing a conference. This is our 3rd conference. Before the pandemic issue, this conference was conducted once in 2 years. But due to the pandemic issue, it was postponed a few times. And this year, we continue again with the idea to conduct or to run this 3rd International Conference, Medical Science Technology, virtually on this coming November. So if you are interested, you have your own project, maybe student or staff, or maybe postgraduate students, you're interested to present virtually, attending this international conference, please come and join us. You can have the details in our website, and that website will be updated from time to time. But if you need further information, you can contact me, or maybe you can contact Dr. Shiha, the one, the PIC that in charge in our program today. Okay, I think that's it from me, and the general idea about the technique and laboratory technique for identifications of virus. So back to our chairperson for today.
Okay, thank you. Thank you, Dr. Azima, for the great presentation. I think it's so clear for us. And
okay, sorry, how to close this one?
Oh, okay.
Okay.
Maybe any button to close the screen?
This one. I think I've shared, stopped the sharing, right? Have I stopped the sharing? Saya dah stop the sharing the
Okay. Okay, maybe.
Sorry, I'm not really familiar with Zoom.
Yeah. My commit want to stop the share screen.
Okay.
Wait for a minute. Wait for a minute.
Okay. So anything?
Okay.
Okay.
Yeah. Okay, thank you, Dr. Azima.
Sorry, can you hear me? I have a problem with my internet connection now. Can you
Hello?
hear me? Can you hear me?
Yeah.
Okay. Sometimes there are some problem with the internet connection. So sorry kalau saya tak berapa dengar.
Oh, enggak apa-apa.
Okay.
Hanya tidak terlihat saja.
Okay.
Okay, thank you. And we gain more of knowledge about virology, like as basic virology, virus component, virus detection, direct and indirect detection, traditional and modern detection, isolation method, and the last information is about international conference.
Yes, yes, international conference. You are welcome, you and your members, your students to join us.
Okay.
Jumpalah.
Inshallah, this information can send to my institution, and I hope my student or the lecturer can join the international conference. Okay. All right. Now we'll move to question and answer session. If any question, all of the participants can raise your hand or write a question in a chat room. Maybe any question? Silakan bisa bertanya, mungkin bisa dengan bahasa Inggris atau bisa dengan bahasa Indonesia.
Boleh saja mana-mana.
Okay, maybe Mas Alif Rahman Devaha can unmute and ask a question.
Thank you so much for the time, Pak Arif. First of all, I would like to thank to Dr. Azima Abdul Wahab for the knowledge that you share with us. Thank you so much. Okay, so earlier you explained it about viral diagnostic, right? So of the two diagnostic between direct and indirect, which one is more recommended for diagnosing viruses? Because each diagnostic has advantages and disadvantages. Is it just adjusting to field condition or adapted to the referrals received, maybe? Thank you so much.
Okay, for me, the direct technique, the direct detection is more suitable for rapid diagnosis because it involves applications of, let's say, immunofluorescence, or you directly detect the nucleic acids from the source of sample. Compared to the conventional method, I mean the indirect detection, it takes a longer time to produce result. It's slow turnaround for you to get the result. So in the situation or scenario of pandemic issue, like a COVID-19 issue, so the best technique or method that we can adapt or adopt for our diagnosis is more to the direct detections.
Okay, thank you so much. I have one more question. So I want to ask, maybe all this time I've only heard about one type of virus in one cell. So I have a question. If there are two viruses during superinfection in same cell, what is the possibility that will happen? Is there a genetic interaction or what? Maybe.
Sorry, can you repeat again?
Okay. Maybe I've only heard about one type of virus in one cell, right? If there are two viruses during superinfection in same cell, what is the possibility that will happen?
Yes, I understand in certain condition like a respiratory tract infection, there's a what we call it co-infection circle. For example, respiratory tract infection, so respiratory syncytial virus infection can co-infect with other types of viruses. So in terms of viral detections, sometimes if they for me if they are from similar family, it's quite difficult for us to distinguish the detections from the same family strains. So yeah, for the culturing, that's the reason why when we need to perform the cell culture technique for the specific types of viruses, we need to do the identification first. What is the best host or the culture system that can provide propagation system for the virus itself? The combination of co-infections by two different viruses need, for me, need further investigation for the confirmation.
Yeah, I see. Okay, thank you so much, Dr. Azima. Saya kembalikan lagi ke Pak Arif.
Okay, thank you for a question, Mas Alif ya. Okay, any question? Dinda Azra Maura. Okay, you can unmute and ask your question.
Thank you for the time given to me, Pak Arif. I have something to discuss about Western blotting to Dr. Azima. She said that we need a really low temperature when we do the Western blotting. So the question is, what is the optimal temperature that we need when we do the Western blotting?
Okay, for that particular question, yes, I have mentioned that when we are handling with a protein sample, everything must be performed under low temperature. So what normally we use here in our research work, every time we handle a protein sample, let's say you you perform the isolations and extractions of protein from sample, and everything must be done on ice. There's no specific, I would say, specific temperature, but everything conducted on ice to ensure that the proteins will not be denatured later on. We need to maintain the conditions of the protein starting from the first part. You handle the proteins, and then you do the SDS page running process. And then when you try to transfer your protein from the gel, SDS gel, onto the PBF membrane, it also must be done under low temperature. We require, if you purchase Western blotting technique, it comes together with a pack of ice that you can use during the transferring process, because at that stage it involves transferring of proteins from gel onto a membrane. So the condition, the environment in the buffer, transferring buffer must be in the low temperature. again to maintain the conditions of the protein, so that later on when you apply and primary and also the secondary antibody, it can directly bind to that specific or targeted proteins on the brain. If you do not apply the low temperature, most probably your protein will be denatured, and then you will get false results later on. Am I answer you?
Okay, it's quite clear, Doctor. Thank you so much for the explanation.
Okay, thank you.
Okay, thank you, Saudari Dinda. Any question from the other? Or any question from a student from Malaysia? Or you can write a question in a chat room. Okay, another question.
Okay, just in case if any of you are interested to know more about the technique, or maybe let's say just now one of the participant asked me about the Western blotting. Yes, you are welcome to contact me. Maybe I'm not sure. Maybe you have the MC or maybe the committee has my email address, so that maybe it can be shared to the student. So if any student have something to ask me, you can directly email me or more information.
Okay, thank you, Dr. Azima, for the information. Any question? Another question? Or okay, Alif Rahman Depa, another question. You can unmute and ask your question.
Okay, thank you so much. One more question for me. So before you mentioned about gel electrophoresis is a standard method to analyze amplified DNA product, right? So I want to ask, why should amplified DNA products be analyzed using gel electrophoresis? Are there any DNA amplified products that fail if they have to be analyzed again using gel electrophoresis?
Okay. The reasons why we need to perform the gel electrophoresis is just to confirm either the product, the product from the amplifications of the PCR is there. If you can see the results, so it indicates the presence of the positive results from your PCR. At the same time, for certain research work, they would like to see the concentration and the concentrations of product from the PCR as well, so it can be detected by the gel electrophoresis.
Okay, thank you so much for the answer.
Okay, one question again. Any question? Dari angkatan 2020 mungkin yang sedang kuliah mengenai tentang virus, bisa dengan bahasa Indonesia juga, tidak apa-apa. Halo? Silakan Mbak Kusnul atau siapa ingin menyampaikan pertanyaan. Oke, mungkin pertanyaan terakhir dari angkatan 2020, apabila ada pertanyaan.
Saya, Pak.
Silakan. Boleh bahasa Indonesia apabila kesulitan.
My name is Latifah Sukma. I want to ask about ada ke virus yang bermanfaat bagi badan manusia sehingga tiada menyebabkan penyakit. Terima kasih.
Okay. Maybe the advantage of virus for our body.
Okay, soalan tu okay, for that kind of question. Okay, basically viruses is known as obligate parasites, meaning that they are categorized as a parasite. Parasite means the interaction between two parties, antara dua host, which one of the party maybe are affected with the interactions. Maksudnya kebanyakan, I would say most of cases the involving virus is related to the infections. But there's one group of virus known as a bacteriophage. So this bacteriophage in terms of the structure, it has a tail, but this bacteriophage have been proven never cause any disease. But it helped scientists untuk membuat kajian, which this bacteriophage can be used to infect bacteria, for example E. coli, so that dia ada application dia sendirilah, penggunaan dia sendiri yang mana dia tidak mendatangkan penyakit, but it help scientists for their infections of organism, especially E. coli. But most of cases when we talk about viruses, they are considered as obligate parasite. Dia boleh menyebabkan penyakit, either in human, animals or plant.
Okay, thank you. Ya, jadi mungkin memang kebanyakan ya untuk virus itu terkait dengan infeksi dan penyakit. Mungkin berbeda dengan mikroorganisme seperti bakteri dan lain-lainnya.
Betul. Kalau dibandingkan dengan bakteria, boleh dikatakan just a few group of bacteria considered as pathogenic, right? Dia boleh menyebabkan penyakit. Tapi the rest of them they have their own beneficial. They ada benefit in terms of the food production, for environmental cleaning process, and a few more.
Like fermentation or
yes, for fermentation process.
Okay, thank you, Dr. Azima.
Okay.
Maybe any question? One question from Dinda Asra. The last question, ya. Dinda Asra, you can unmute.
Thank you, Pak Arif. There's one more question from me about immunoassay non-competitive. If in non-competitive immunoassay enzyme, if the negative control gives the color, what is the cause of the presence of the color? Thank you.
As I mentioned just now, the results, the interpretations result for the competitive is inversely proportional to the finding, meaning that if we can find or detect the results or signals, it will indicate. How can I explain? It's quite difficult for me to explain for the competitive compared to the direct and indirect. Because it's a competition, the competitive name actually derived from the competition between antigen in a sample and also target proteins on the ELISA plates. So if there is a binding between a protein and is there a binding between antigen in a sample to the label antibody that you have added together in the process, and then they will remove or wash away later on, dia akan tinggalkan sakit-sakit that can be detected by the system. So tadi, if you receive positive results, meaning that positive result, you can still detect the signal, meaning that your sample most probably doesn't contain any targeted protein. Okay. Okay, that's it.
Okay, thank you. I think So it's enough and so clear for us. Okay, before we close the webinar, as a moderator and representing Universitas Aisyah Yogyakarta, I would like to submit a certificate of appreciation for Dr. Azima as a speaker. Maybe panitia bisa menunjukkan. Maybe committee can share screen for certificate appreciation for speaker. Okay, thank you.
Okay, thank you so much.
Dr. Azima for the time and for more experience and more knowledge for us. And I hope we can meet again in a next moment.
Inshallah.
Inshallah.
Inshallah. I'm also happy. Thank you so much for the invitation. Saya seronok with the collaboration when we have this online lecture. Inshallah in the future, I think in the next few weeks, we are going to have same session with Dr. Nisha for the molecular diagnostic in parasitology. Maybe you'll see another technique for detections of parasites. And then inshallah kalau ada rezeki, maybe next time we will invite maybe some of your lecturers to share your knowledge and also to share maybe your some experience in the research area. Okay.
Okay, thank you for Dr. Azima. Okay, finally we have the end of this webinar. I would like to say thank you to Dr. Azima for the great presentation and the great topic, and then the audience for their active participation and the more question, and I think it's good discuss for us. Thank you for the attention. I think it's enough. Saya tutup. Wassalamualaikum warahmatullahi wabarakatuh. Waktu dan tempat saya persilahkan kepada Mbak Kamel selaku MC. Thank you, Dr. Azima.
Okay, thank you so much kepada Encik Arif.
Terima kasih kepada dokter. Thank you very much for the excellent presentation and discussion, so that we can get a lot of provision in preparing ourselves to become medical laboratory technology expert. Terima kasih banyak atas pemaparan dan diskusi yang luar biasa, sehingga kita bisa mendapatkan banyak bekal dalam mempersiapkan diri menjadi ahli teknologi laboratorium medis. Selanjutnya, penyerahan sertifikat kepada moderator, Bapak Arif Yusuf Wicaksana. Dipersilakan kepada admin untuk menampilkan sertifikatnya. Untuk operator, apakah masih terhubung sehingga bisa menampilkan sertifikat yang akan diserahkan kepada moderator? Baik, terima kasih. Sertifikat ini kami serahkan kepada Bapak Arif Yusuf Wicaksana yang sudah membantu jalannya diskusi dan pemaparan materi dari Dr. Azima. Kepada Bapak, mohon diterima.
Oke, terima kasih untuk Mbak Kamel atas sertifikatnya. Sekaligus memberikan izin untuk saya sedang posisi share absensi ya untuk peserta yang hadir, baik dari Malaysia ataupun dari Indonesia. Mohon maaf terlambat. Oke, terima kasih Mbak Kamel.
Baik Pak, terima kasih kembali. Kita lanjut. Sebelum mengakhiri acara pada sore hari ini, izinkan kami mendokumentasi kegiatan. Dimohon kepada Bapak, Ibu, dan mahasiswa untuk menyalakan kamera Zoom. Dimohon kepada mahasiswa baik dari Universitas Aisyah maupun dari Universitas Kuala Lumpur untuk menyalakan kameranya agar bisa mendapatkan dokumentasi bersama. Ditunggu ya teman-teman untuk menyalakan kamera Zoom-nya. Baik, terima kasih kepada rekan-rekan yang sudah menyalakan kamera Zoom-nya, maaf. Pada hitungan ketiga, admin akan memfoto layar yang ada pada saat ini. Untuk slide pertama, 1, 2, 3. Baik, terima kasih. Selanjutnya slide kedua, saya hitung lagi. 1, 2, 3. Diinformasikan ini sampai slide ke-7 ya teman-teman. Baik, untuk slide ketiga, 1, 2, 3. Selanjutnya slide keempat, 1, 2, 3. Selanjutnya slide kelima, 1, 2, 3. Selanjutnya slide keenam, 1, 2, 3. Dan yang terakhir slide ke-7, 1, 2, 3. Oke, baik, terima kasih. Terima kasih juga kepada operator. Thank you very much to the presenter, moderator, and participant who have listened to the event from beginning to end. Terima kasih banyak kepada pemateri, moderator, serta para peserta yang telah menyimak acara dari awal sampai akhir. Saya mewakili panitia yang bertugas menyampaikan mohon maaf atas salah kata dan perbuatan. Akhir kata, billahi fisabilil haq fastabiqul khairat. Wassalamualaikum warahmatullahi wabarakatuh. Terima kasih banyak.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you so much. Ya.
Oke, terima kasih semua.
Ya, Bu. Untuk rekan-rekan dipersilakan meninggalkan via Zoom.
otot kawan tulang besi.